Russ is an author on this work, as well as Wei Chen. This is work that we've been doing in my lab. This sounds really loud, so I'll try and talk a little softer. So our goal is extracting information. The idea is we live in this age of bioinformatics, and problem is, up until recently, it's been more informatics than biology. And the work we try and do is trying to think about how do we use our understanding of biological systems, or really hypotheses about different biological systems, and fit them to data. Okay, and the data we're dealing with is genomic data, and I'll go into it in a little bit. The idea is we're going to extract information on the protein translation process. So we're going to think about errors and costs of pausing, okay, that occur during protein translation. We want to learn more about that, and we're going to try and get that understanding by looking at patterning within genome sequences. Okay. All right, so I'm going to try and go quickly on these slides, but just we're evolution. I'm an evolutionary biologist. Evolution is how we make things. Evolution is how we make sense of things in biology, so I like this work because it allows us to make sense of genomic data sets, and the key thing is it uses principles of population genetics to do it. So it's not only evolutionary biology, but it's put in this very old formal framework of population genetics, and it's just awesome as a result. One of the things that we do, other thing I like about this work, is that we actually take the idea that Sewell Wright presented in the 1930s about a fitness landscape, and we show how we can define a fitness landscape. And we actually have an adaptive landscape, which describes how adaptive a trait is, and then we link that to a fitness landscape. And then we use the concept of fitness landscapes, and we look where populations are on those fitness landscapes to be able to make inferences about the biology going on. Okay, so we're able to sort of parameterize and formalize these ideas that most of the time when we run into as grad students, we're just like, oh, that's really cool, but high dimensional spaces. But here we actually really use these concepts. Okay. Okay, so the goal of the work in my lab is to take models of how we think proteins are assembled, or the protein translation process. We're going to think back to your genetics course of ribosomes moving along mRNAs. And we're going to think about subtleties and errors that can occur and costs that can occur during that process, and try and formalize those into mathematical models. And we're going to take those models that think about the costs and benefits of protein assembly, and we're going to put them in the context of an evolutionary process, and so we're going to think about the evolution of these sequences under different selection regimes. Okay, and the idea is that we're going to try and extract this information about these things by using models that describe these things. One of the key things we get out of this work is we get the ability to take a genome and we can predict the protein production rates of a gene based on the pattern of codon usage within that gene. And we find that actually our predicted rates are on par in terms of the predictability of empirical estimates of these rates. So we're quite happy with that. And we also gain insight into understanding some and really documenting some of the more complex behavior of how codon usage changes as we change gene expression. So if you talk to most people who think about sequence evolution, they're all very aware codon usage changes with gene expression. But what we see with our models is that these changes are actually nonlinear in behavior, and that nonlinear behavior comes very naturally out of the models we use. And nobody's really ever talked about this. Everybody just thinks these things increase monotonically. And we actually see that the models predict non-monotonic behavior, and the data is real consistent with that. Like, why are we doing this work? Well, because it does math and it does statistics and it's biology, and we think it's really cool. The other reason why is because really we're at a point where sequencing genomes is becoming a very, very easy way to gather biological data. And we know that biological data has information in it, but we don't necessarily know how to pull that out. And so the idea is by taking biological models and applying them to this data, we can pull that information out. Okay, so just to give you a little background on where our models are coming from, for those who haven't taken a genetics course in a while. Remember DNA, we've got 4 letters in the DNA alphabet, A, T, G, and C. Proteins are going to use, essentially, there's 20 canonical amino acids that are used, okay? So, and there's also, if we think about it, there's 20 canonical amino acids, and then there's a stop signal. So we have essentially like 21 signals that we want to give, but the structure of the genetic code is such that we have 64 different individual signals we could give, Okay, so there's an inherent redundancy because we essentially on average a 3-fold redundancy in the genetic code in that we really only need 21 signals to be sent, but we have 64 different codons that could be used. Okay? And so that redundancy means that certain codons, like there are 2 amino acids, methionine and tryptophan, that only use 1 codon. Okay, so 2 out of the 20, there's no codon use, there's no sort of variation in, or multiple synonyms for codons. But for everything else, we've got at least 2 codons that we can use to put in that amino acid. Okay? And what we find is that there's information. This sort of gives biology a degree of freedom to encode information into it. And so again, what we're trying to do is build models to extract that information. Right? So we see that about half of the amino acids use 2 different codons. So we have tyrosine, histidine, glutamic acid, aspartate. I can't remember all of them. So anyways, we'll just move on. Okay? So we have most of them are using 2, but there are even some that use 6 different codons. Okay? And so we have a wide range of redundancy. So I'm going to look at glutamic acid, which can be encoded with GAA or GAG. Okay? So if we look at like the yeast genome and we go to every gene and we say when it's putting in a glutamic acid, what is the frequency it uses GAA versus GAG? Okay? And if we had a simple kind of null model where if we assumed there was no mutation bias and there was no difference in the efficacy of these different codons, we would expect to see a distribution of codon usage of GAA across the yeast genome, and I'm using here as an example, to follow a distribution that sort of looks like this. Okay? But if we actually look at the yeast genome, we see actually it's biased towards using GAA more frequently than GAG. Okay? And indeed, there are some genes that use GAA exclusively. Okay? Yeah, there are also some that use GAG exclusively, but there's very few of those. Okay? But we see this deviation from sort of uniform usage. Okay? And that's what we call codon usage bias. So it's non-uniform usage of synonymous codons within a gene. And Crick actually predicted we might see something like this before we even began sequencing genes. And the first genes that people began sequencing were mRNA sequences of ribosomal genes, because those were in very high abundances. And lo and behold, we saw that those genes were very biased in which codons they used. Yes?
Quick, stupid question. So that's frequency within a gene?
Within a gene. So this is like 5,000 genes sort of all together. Okay? That was a really good question. It's not stupid at all. All right? Okay, so the idea is that if this deviation from uniform usage is being caused by some kind of process, if we can build different models and we can fit them and to see are those different models, how are they consistent with this deviation, and what parameters are consistent. So we can have different models, we can do model comparison, we can also do parameter estimation about what could be driving this. Okay? So there's a number of different explanations out there in the literature about what can be responsible for this. And none of these are mutually exclusive. Okay, we're focusing on one subset, but it's a simplification of the world. All of these are probably contributing, but we're focusing on a subset that we think is probably very important. So part of this is, this deviation is due to the fact that mutation between, this is GAA and GAG frequency, so the mutation from A to G isn't necessarily equal from G to A. And that bias can lead to a deviation from a 50-50 expectation. Drift itself sort of lends variance in this whole process. Okay? And so I think of those as sort of non-adaptive processes. And then if we think about adaptive processes, 3 of the most commonly invoked ones are that these different codon usages have to do with things like mRNA stability. Choosing different codons leads to different levels of mRNA stability, and mRNA stability can affect a number of different things about initiation of translation or the half-life in the mRNA. Okay? Packaging of the DNA itself can be affected by this. Okay, and we're going to focus, though, on thinking about the efficiency of protein translation, because what we find is that there's a strong relationship between how often a gene is expressed, and I'll talk about what I mean by gene expression in a second, and how much bias we see in that gene. Okay, and that's consistent with the idea that protein translation, selection for efficient protein translation is driving this. Okay, so in our work, we think about the source of natural selection on stemming from efficiency in protein translation. We ultimately are able to calculate, thinking about a gene, we can think about what we call its translation cost-benefit ratio. So it's essentially thinking about how many ATPs, either directly or indirectly, is the cell going to have to expend to get one peptide. Okay, and so we think about the cost of ATPs, and then we also think about how functional is that peptide likely to be in comparison to a complete peptide without any errors. Okay, so we avoid the difficulty of thinking, what does the protein really do? And the hard things about protein folding at this point and protein functionality, we just sort of think of things relative to an error-free protein. Okay, and so our assumption is that natural selection is going to favor this cost-benefit ratio, minimizing this cost-benefit ratio, which we represent with an eta. And the idea is then that the strength of selection is going to increase with the target protein production rate of that gene. So having a very low cost-benefit ratio isn't going to matter too much if the gene's being produced once an hour. But if you're producing thousands of those proteins from that gene a minute, then reducing that cost-benefit ratio is going to have a strong fitness effect. Okay? And so we'll formalize all of that, but that's kind of the intuition. So again, what we're thinking about now is what's going on within the ribosome as it's assembling the protein. We're going to be primarily focused in this work, we're going to be talking about the cost of this ribosome pausing, looking for that correct tRNA to match that synonyms, because this different synonyms can have different efficiencies with which it matches and translates forward. Yes?
Is that scaled to the length of the protein?
We calculate as a function of the entire length of the protein. So it is a function of the length, but we don't divide it by the total length. Sometimes we'll divide functionality by the total length, thinking that each component, if the length is n, then each position sort of contributes 1 over n units to functionality. Okay? But in some sense, the way we try and build these models is that the length naturally is embedded within the model itself. Okay? Yeah, if you ask Russ, I'm really anally attentive about trying to think about, making the biological properties emergent from the model itself as opposed to having to kind of impose them. Okay, so ways of thinking about increasing translational efficiency. One of the ways, and this is the most commonly invoked source of selection for translational efficiency. It's not necessarily the strongest force, but it's the one that people invoke the most when they're giving explanations. Is that different codons have different probabilities of putting in the wrong amino acid. Okay, and so that their selection to minimize missense errors. We're exploring this, but we're not totally sold on this because a lot of times a missense error has very small effect on protein functionality. You can change 50% of a protein, you can probably change the amino acid and not really affect protein function much at all. Okay? Another way of increasing the translational efficiency of a gene is to use codons that have a lower probability of a nonsense error, of the ribosome terminating translation before it gets to the end. Okay? And we can imagine that if you stop translation before you get to the end, so if somewhere in the middle or just somewhere in the middle or so, that the functionality of the protein you're going to get is going to be probably 0. Okay, and so this is where we started the work because it was really easy to map function to errors, because you'd be like, well, if you stop before you get to the end, we'll assume it's 0. You had a question?
Can you think about the genome architecture and where these things are? So there are these hypotheses about closer to centromeres, closer to telomeres, and whether there would be an effect of the rate of...
No, we don't, and we could conceivably put that information into the model. Well, we're doing like a first-order approximation in some sense of these processes. Adding those types of things in there once we have this worked out are what we have looking ahead. We would
necessarily a good idea. I was just wondering.
Well, no, actually, I think it is. I mean, I think the idea is we can begin asking hypotheses about, like, there's likely the mutation rate and the bias in mutation can likely vary between different positions in the genome. So can we take that into account? Okay, yeah, we can, and we can try and estimate those types of forces. But right now we're keeping things really simple. We're not looking at the architecture, overall architecture itself. Okay? But I think those are fair things, right? And the thing is, the argument of why being so anal retentive about these models is useful is because we're so explicit about the biology. We can always add. We know exactly where to put more biology in because it's easy to see where to put it. So what I'm trying to talk about today is this is the simplest model we can use that we use, and it's thinking about selection on different synonymous codons to minimize the waiting time the ribosome spends looking for the correct tRNA. Okay? And so we call this our ribosome overhead cost model, and I'll sometimes refer to it as this ribosome waiting time model. And the idea is that natural selection is favoring synonyms that minimize the amount of time. This giant factory, ribosomes are big, expensive things to make with a limited lifespan. How much time they're spending there churning through tRNAs looking for one that stably matches before they can move forward. Okay? Because if they have to spend a lot of time looking, that's sort of wasted potential productivity that they could have. Okay, so where does these costs from? Well, the ribosomes are expensive to create. They're big. They're made of proteins and a lot of RNA. Okay, they're going to have a limited lifespan, and there's finite numbers in the cell. All things that are easily sort of defensible assumptions. Okay, and so what we do is this pausing time model. What we're going to do is we're going to think that the ribosome has this cost of assembly, and it has a limited lifespan. And so we're going to think that the cost of pausing is going to be related to how long, essentially, it spends pausing. Okay, so or the cost of protein assembly is going to be related to how long it spends pausing. We also implicitly are including the direct cost of protein assembly, but that cost isn't going to vary from one set of synonyms to the next, because if I'm going to make a protein and I don't have any errors, if it's n amino acids long, I'm going to use essentially 4 times n amino acids to synthesize it directly. What we're thinking about is, and it wouldn't matter which synonyms I use, what we're thinking about is the idea that different synonyms mean that it has to pause different amounts of times, and so that indirect cost of its pausing varies between different sets of synonyms. Okay? I'm getting some nods, so I'll take that as I can move ahead. Right? And so we have this eta term here. And so we're thinking here, we have this idea of linking, okay, the choice of codons in some sequence I. So that's what this X is representing. It's representing sort of the counts of different codons in a sequence. Okay, and it's given a set of parameters, and these parameters are these T's here are related to, if I use that codon, how long on average will a ribosome spend pausing looking for a good match? Okay, so in our model here we have this X, which is describing the codon counts. This is in gene I, and then this is codon J. Tj is the pausing time of that codon. So we essentially do is we look at a gene sequence, or we be given a gene sequence. We look at the different codons, and we add up how many times do I use this codon in the sequence, how many times do I use that, and I scale them by their waiting times, and I just add them all up, and that gives me an idea of how much pausing time do I have for that synonym versus another synonym. Okay, and there's some scaling constants. I'm not going to talk too much about them, but this K here sort of describes how much ATP am I essentially spending for every second that a ribosome spends pausing looking for a tRNA. It's just a scaling term. We'll see a few others. Okay. So I mentioned this before. We're going to assume that the strength of selection is going to be related to the expression level of the gene. Okay, and so selection favors alleles with lower cost-benefit ratios, or production costs, these eta terms. Okay, and the idea that the strength of selection increases with the gene's average protein production rate. So again, something that comes out of this model. If you read the codon usage literature, you find people talk about gene expression, but they're not usually very explicit about what they mean. If you realize you're thinking about protein translation, then when we talk about gene expression, we mean the average protein production rate of that gene weighted across the lifespan of the organism and its environments. Okay, so if you were sort of to think about, and I was going to go grab an organism randomly out in the environment, it's my expectation for how often they're making that protein at that moment in time. Okay? And the idea is that we're going to think about selection being related to how many ATPs per second is the organism need to spend to meet its target production rate, which we're going to represent as phi, so that can vary between genes, multiplied by how much energy does it cost to get 1 unit of phi, which is what that cost benefit is describing. Okay, so this phi is saying how many ATPs do I have to spend to get one functional unit of that protein, and then phi is saying on average how often does the organism want to build that a functional unit. Okay, and so if you think about this, this is sort of the average amount of energy an organism spends to satisfy that gene requirement. Okay, and we then assume that fitness is going to, for every ATP you can save per second, we're going to assume you get some small incremental gain in fitness. Okay, and if we assume that, that leads naturally to an exponential relationship between this production energy use, the production rate, times the production cost, multiplied by another scaling term here, okay, and raised to the e. Right? And so the idea is that every time I can say 1 ATP per second, then I'm going to get some small q, e to the q gain in my fitness. Okay? And so we think about there's a minimum product cost-benefit ratio. So this would be the most efficient way to make the protein. Here's the most expensive way to make the protein. There's an average in here. And the idea is that depending on my protein production rate, fitness can drop very quickly or slowly, depending if I'm high expression. As I move away from this minimal cost, fitness drops quickly. As I'm lower expression, it drops more slowly. If phi was 0, it wouldn't matter what I did, right? Because there's no cost, because it's the beauty of 0. Okay, so we can take that now. So we've gone from genotype to phenotype, which is the cost-benefit ratio, and then we've thought about protein production rate, which then allows us to go from phenotype, put it in and relate it to energy use, and then we think about energy use and fitness. So we've gone from genotype to phenotype to fitness, which to me is really cool. Okay, and then I can go into models of population genetics, and I can think about what's the probability I would see a given sequence. Okay, if I went and looked out and grabbed an organism, I can ask what's the stationary probability I would see something with that cost-benefit function given a set of parameters and given a production rate. Okay, and that just comes from models in population genetics. We're using a specific right, essentially derived this, but then Sell and Hirsch explained it in a way I could really understand. And it's essentially like a Boltzmann distribution where the fitness is related to is sort of related to the inverse of the energy of a state, and effective population size is like related to inverse temperature. Okay, so as effective population size becomes infinitely large, it's like taking a physical system and bringing it down to absolute 0. Effective population size is infinitely large, we're only going to expect to see it at the optimum. Okay, effective population size gets really small, it's like raising the temperature in a physical system, you could be almost anywhere. Okay, it's equivalently, if I change phi, I bring phi to 0, you could be anywhere. Phi becomes really large, selection becomes really strong. Okay. So we can now have this ability to take a series of codon sequences, okay? And we can even take, so these are, thinking about this as gene 1, gene 2, all the way to gene N across a genome. And then if we have ideas about what their protein production rates are, we can calculate, we can take this probability statement we have, and we can generate a likelihood statement, and we can make estimates about what the different pausing times are and what the different mutation biases are, okay? And so this is work I did with a graduate student of mine a number of years back, where we went and we looked at the yeast genome and we fitted our model to the codon usage in the yeast genome. Okay, so we took the yeast genome and we took all 5,000 genes and we put them in small little bins. Okay, we calculated the codon usage, like this phenylalanine. How frequently did it use TTT versus TTC? We put them in bins based on their average production rate. Okay, and so we have the dot, which is like the mean in that bin, and then we have like 2 standard deviations around it. Okay, and we see things over here. This is low expression here. We see this, we attribute to just differences in the mutation bias. So the mutation rate, this implies the mutation rate from C to T is substantially higher than from T to C. And that the variation in codon usage here can be explained to just mutation alone, because phi is really small and selection's not having a very big effect. But as we increase the selection, as we increase the production rate, selection begins to affect the probability we see a particular codon, okay? And we see a swap where at very high expression we expect to see TTC, okay, where the selection has essentially overwhelmed the mutation bias, and we don't expect to see any TTT. Right? And we can see these kinds of beautiful little curves as we look at different amino acids. Okay? And we have this Boltzmann distribution, and what's the issue that's going to happen when we move ahead a little bit is calculating this denominator. When we know gene expression, it kind of becomes easy to calculate this denominator, but when we don't know gene expression, then it's sort of like trying to do a Boltzmann distribution, but you don't know temperature, okay? Which makes things hard. Okay? And that's when things become computationally very intensive. Okay, here are just some other curves. I don't want to spend too much time with them. I just want to show that we have some really interesting nonlinear dynamics here, and we can explain it due to these balances of selection and mutation. Okay, and again, people have thought about these forces driving codon usage bias, but nobody has ever really thought about the fact that you can have a gene that's favored by mutation and is like the second most efficient codon. So you can have a codon that's favored by mutation. It's the second most efficient, and it will actually increase in frequency of usage, but then later on drop. Okay, and that kind of nonlinear changes to me makes my heart kind of warm. Okay, the expectation function we're using here in some sense is quite simple when we're thinking about individual sites, and we're looking at one amino acid at a time. It's a Boltzmann distribution, but we can also think of it as a multinomial distribution, that there's these probabilities of seeing one of 2 codons, or one of 3 codons, or one of 4 codons. Okay, and we're thinking about those. We can think about those as samples from a multinomial. Okay, and so here is this again is this work. I thought I had the citation there. This is from my graduate student Premal Shah, who's now working with Josh Plotkin up at UPenn. From a couple years back, and we analyzed the whole genome, and it get really pretty pictures. Okay? But when we did this analysis, right, we assumed we knew the protein production rates of the genes. Okay? So we only had to we estimated those pausing time parameters and those mutation parameters, but we assumed we knew expression rate. And that made things computationally relatively straightforward. Okay? So the thing is that this is changing a bit because our techniques for sequencing are improving time and time again. Okay? So we have to get this out before it becomes obsolete. It's not quite true. But the thing is that we often we have these genome sequences, but we don't necessarily have good gene expression databases. In some sense, sometimes for these microbes, we don't even know under really what conditions do they grow well. We're able now beginning to sequence things, but we can't even grow them. Right? The idea is that if we can look at their genome and we can figure out what the expression rates of different sets of genes are, that will actually tell us a lot about the biology. Okay, because we know, oh, this one spends a lot of time in an anoxic condition, and this one spends a lot of time in anoxic conditions, and oh, this one's a lot of UV radiation, this one not. Yes?
For your modeling, is most of the data that you use, is it microarray data?
It's, yeah, so when we did the fitting, it was microarray, mostly Affymetrix and some NIMBioS data, and what we want is absolute expression values for that work. But what we're doing now is we're going to show, and Wei Chen will talk about the mechanics, is we're going to show how we can fit these models now without even having expression data. Okay, and then we can show, we can compare, we can do fits, and we can do predictions of gene expression, and they're in line with what people measure with microarrays. Okay. 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 So, what we're going to do, the latest change we've done is we've moved the need for this, the estimates of the protein production rate, this vector of phi values, one for every gene, okay, and we've begun treating that phi as an unknown random variable, okay. So it becomes one more thing, and if you think about it, it's like before we had about 40 parameters for pausing time, and we had about 40 parameters for mutation bias. Okay, and we give me a genome and 5,000 expression levels, and you can see how estimating 80 parameters wouldn't be so hard. Okay, I'm now trying to just use a genome. I'm going to estimate those 40 and 40, so 80 parameters. But then I'm also going to estimate another 5,000 parameters, one expression level for every gene. And you might be like, oh, you've got your model over parameterized, and I'm going to be like, no, we don't, because you think about it, the average length of a gene is about 400 amino acids. So I have like 400 observations for every gene, okay? So getting one parameter per gene isn't actually going to be, we believe, and our work shows that the information is encoded in there, and our models, we don't have to worry about over-parameterization. We're really taking the approach that Wallace et al. Published last year, where they began thinking about error in the protein production rate. So in our work with my graduate student Primo, we assumed we knew protein production rate exactly. And they sort of, Wallace is a very nice paper, and they talk about how that affects your estimates of selection, okay? And we also realized we'd been looking for a while for the hammer to solve this problem that I'm describing, and we realized we could take Wallace's approach and just expand it a little bit and begin not assuming there's no noise in our estimates of selection, but there's no estimates of production at all, and just start treating that as a completely random variable where we don't have any observations. Okay? So we're essentially taking Wallace's approach where they assumed there were some observations that was log normally distributed around some true values, okay? And we've gotten rid of this need of those observations. Okay, and we have this method now where we're going to estimate protein production rate. We're also going to estimate these mutation and pausing time parameters using just the genome sequence itself. Okay, and so what do we get? Well, we run these MCs for a long time on these computers and move a lot of electrons around. And so what I have here is this is looking at the posterior samples. And when we're doing these MCMCs, we're sampling in the protein production rates for every gene. We're doing sort of a random walk with rejection sampling, and we're also doing like a Gibbs sampling approach for the pausing times and the mutation rates. And so we run these, and we get samples from the posteriors. And so what I have here, these are estimates from an empirical study where they use microarray data, Affymetrix chips from Yasour et al. Like 2009, and this is their measured mRNA abundances, which people think is related to protein production rate, and then this is our posterior estimates of those protein production rates. And we get an R squared of about 0.5, so 50%, we're explaining about 50% of the noise, but that means our correlation coefficient is about 0.7, okay? And if you look at between labs, the correlation coefficient between different microarray studies between labs is on the order of 0.7. Like within a lab, they can be really high consistency, but there's all these systematic variation between the different things. So we're very happy with this. Okay. If we take the technique that from Shaw and Gilchrist, and then that was expanded by Wallace et al., where we assume we know expression level, or at least we have noisy or even noisy estimates of expression level, and we use the noisy estimates of expression level, and we integrate that information with the codon usage bias and the fitting of the model. Okay, so it's sort of like it's the same model we're using, but here we're we're using that Yasour data to fit our model. Okay, and here we're estimating the protein production rate without using the Yasour data. Okay, we get a very, very high correlation coefficient, about 98%, which sort of to us tells us that even that Wallace approach, where they said, oh, we know expression level and it's noisy, really most of the information is actually coming from the genome itself. Okay, because we have a correlation coefficient of 98%, where here's using phi and here's not using estimates of phi, and they're really tight. Okay? So it says Wallace's information is primarily coming from the genome and the codon usage bias. Okay? This is a comparison. These are the estimates of the pausing times from the Wallace approach, where you have estimates of protein production rate. And these are estimates of the pausing time that we're using where we don't have estimates of protein production rate. What you see is there's this beautiful correlation coefficient of close to 1. Okay? The one issue is that it's a bit biased. Okay, so we're underestimating things, and this has to do with the fact we have an identifiability problem. We're struggling right now to kind of come up with a simple way to kind of explain this bias. But the key point here is that we can really estimate those parameters that Wallace had been and Gilchrist and Shaw had been estimating now without even having a genome. Okay, and we see also good correlations with mutation rates. Okay. We also can go and we can look at we can compare our predictions of how codon usage will change with gene expression. And so this is our estimates of gene expression from our model fit, and then this is the predicted changes in gene expression, changes in codon usage with gene expression. And we see essentially these pictures look almost exactly the same as when we had the gene expression data sets, okay, which says, again, that codon usage patterns have enormous amount of data, essentially is the same amount as if I went out and measured expression levels in the lab. Okay, and so the pictures are very pretty. So what are we doing? Well, we think we have that problem of systematic bias. So now we're moving forward. We're trying to explore more flexible prior distributions for phi. Okay, and we're also looking at approaches so that we can include these other forces of selection, not just thinking about pausing time, but thinking about what is the effect if you put the wrong amino acid in there, okay, and what is the effect is if you terminate translation prematurely, because those models are not mutually exclusive. And so what we're doing is we're trying to build. We're trying to take our models from those. Some of them we're doing some first-order approximations. Other ones we're exploring some new techniques. The idea is we can take that same fitting approach and calculate missense errors and nonsense errors by just looking at the genome. So instead of thinking about pausing time, saying, okay, let's now take these different formulations, biological motivations, and come up with estimates of what the missense error rates are per codon. Okay, it's something that has been measured, but only for a few codons in the yeast genome, and estimate what the nonsense error rates are. And then, because we have a very explicit model, we can, in the end, combine all these three forces together into one model, and so we can find out the relative contribution of these different factors. Okay, so as I mentioned, this early work was done in collaboration with Premal Shah, who's at UPenn. Also, we got a lot of help from the RDAV group at ORNL in developing these first passes at these newer models. We've gotten support through NIMBioS, and we've also gotten support through NSF. And I've talked longer than I wanted, but I'm happy to take any questions. Yes? Essentially, our model is a logistic model.
Okay.
Yep. And yep. And so, yeah, it's a logistic model. And those are the curves and those models. The parameters in that logistic model are delta. Like the intercept at if this wasn't log scaled, because we're fitting it, the data itself isn't, when the logistic model isn't log distributed, but the intercepts in that logistic model, the zero intercepts are the mutation biases. Okay, and the change in the probability of seeing an event, those are essentially as I change this variable, those are the delta t's, or the pausing time scale those. Okay, so it's a logistic model, but it's derived, it's linked directly to biology. Okay, well, thank you very much.